Hi everyone, it's Jessica again, Global Technical Marketing Lead at Croda Pharma. Welcome back to our YouTube channel. In this video, we're going to demonstrate how the excipients you choose directly impact API performance, and how formulating with a higher purity excipient leads to better small molecule drug stability. Super refined excipients can enhance the stability of many small molecule APIs. Our scientists evaluated API stability in both individual excipients and complete formulations to see how purity impacts performance. Today we'll walk you through stability data for the 6 different small molecule drugs here on the screen, each formulated in different excipients such as polysorbates, PEGs, oils, and more. So without further ado, let's take a look at the data.
Hi, I'm Will Small, Research and Technology Specialist at Croda Pharma. Let's kick things off with docetaxel, a widely used chemotherapy drug that is often formulated with polysorbate 80. In the slides ahead, I'll walk you through how using our super refined excipients can make a big difference in improving the stability of this sensitive API. Docetaxel is a widely used chemotherapeutic agent from the taxane family, closely related to other taxanes such as paclitaxel. It's sparingly soluble in water, so intravenous formulations require excipients like polysorbates to solubilize the API. However, docetaxel is chemically unstable in polysorbate 80, which is commonly used in both the originator and multiple generic formulations. So we wanted to investigate whether our super refined versions of these excipients could minimize that degradation and improve formulation stability. To assess this, we ran a 12-week accelerated stability study at 40 degrees C. Docetaxel was solubilized in super refined and compendial grades of polysorbate 80 and also PEG 400, which is similar to the PEG 300 that is also used in some docetaxel formulations. We then monitored drug recovery over time using high-performance liquid chromatography. The results were clear. Docetaxel recovery was significantly higher in both super refined excipients, maintaining over 90% of the API after 12 weeks. By contrast, the standard grades showed considerable degradation, with some batches resulting in well below 50% recovery. These findings highlight the superior stability offered by the super refined grades. Next, we looked at the chromatographic profiles. Docetaxel elutes as a single peak at 3.8 minutes. In the super refined polysorbate 80 and PEG 400, we saw only this peak, even after 12 weeks at 40 degrees C. But in the standard compendial grades, we observed several additional peaks, indicating the formation of multiple degradation products. This provides further evidence that our super refined excipients improve the stability of the API and prevent formation of API degradation products over time. We then used LCMS to determine those degradation products. 3 key compounds emerged: 10-oxodocetaxel, 7-epidocetaxel, and 7-epi-10-oxodocetaxel, all forming from docetaxel in the compendial excipients. Importantly, 7-epidocetaxel is an epimer that's less efficacious than the parent compound and has shown reduced cytotoxicity toward leukemia cells. And none of these degradation products were detected in our super refined excipients. Finally, we assessed the stability of docetaxel in its formulation with polysorbate 80, ethanol, and citric acid. We believed that citric acid was being added to stabilize the docetaxel in the formulation, so we evaluated formulations with this present and also where it was omitted. In the compendial formulation without citric acid, we observed degradation to both the epimer and its oxidation product. But in the super refined formulation, degradation was not observed, even without citric acid. This suggests that our super refined polysorbate 80 inherently stabilizes the API, reducing the need for additional stabilizers. It's a compelling example of how excipient purity can directly impact formulation performance and patient outcomes. Now let's take a look at etoposide, another widely used chemotherapy drug that presents some unique formulation challenges. We'll explore how our super refined excipients can make a significant difference in its stability, just as we saw with docetaxel. Etoposide is commonly used to treat ovarian cancer, leukemia, and other cancers. It's poorly soluble in water, and so it typically requires solubilizing excipients like polysorbate 80 and PEG 300 in its formulation for intravenous infusion. In our internal screening studies, we found that etoposide is unstable in standard compendial polysorbate 80. So, we set out to compare its performance in our superrefined excipients versus the compendial alternatives. Recovery rates were significantly higher in the superrefined versions. In superrefined polysorbate 80, recovery was nearly 100%, compared to anywhere between 17 and 85% in the standard compendial grades. In PEG 300, stability in the superrefined grade was significantly better than in the standard compendial grade, where recovery after 12 weeks had dropped below 30%. The chromatograms at each time point support the excellent recovery observed in the superrefined excipients, with only a single peak present representing etoposide. But in the standard compendial grades of the excipients, an additional peak was observed to form at 4 weeks, which then increased at 8 and again at 12 weeks. This second peak suggests a degradation product, and although we only show the data here for polysorbate 80, the same was observed in PEG 300 as well. As we did in the docetaxel study, we used LCMS to investigate the nature of the second peak and degradation product observed in the standard compendial excipient grades. We confirmed that peak as being cis-etoposide, which is a biologically inactive degradation product formed by epimerization, where the parent API undergoes a conformational change in structure. This was only observed in the standard excipient grades and highlights how superrefined excipients can stabilize the API, preventing degradation and maintaining efficacy.
Hi, I'm Natasha, Research and Technology Manager at Croda Pharma. Now, let's take a look at how superrefined excipients improve the stability of ritonavir. Ritonavir is a water-insoluble antiretroviral used to treat HIV and AIDS. It acts as a HIV protease inhibitor, and while it can be used alone, has been shown to be more effective in combination with other antivirals. It's typically delivered orally, formulated as a tablet, an oral solution, as well as being formulated in a liquid oral capsule using oleic acid. It's known to be unstable in standard purity grades of compendial excipients. To investigate this, we ran a side-by-side comparison using superrefined oleic acid versus the standard purity grade. After 12 weeks at 40 degrees C, the percentage recovery of ritonavir in superrefined oleic acid was more than double that of the standard grade oleic acid, 55% versus 24% API recovery. So while degradation is experienced in both products, the efficacy of the API in superrefined excipients is twice that of the compendial grade. We also saw noticeably fewer degradation products in the superrefined grade, with cleaner chromatograms and smaller impurity peaks. This shows that the higher purity of the superrefined excipient directly improves the API stability in this challenging formulation. Now, let's take a look at haloperidol decanoate, an antipsychotic used in long-acting injectable treatments for schizophrenia. Haloperidol decanoate is delivered intramuscularly as a slow and sustained released formulation. Commercial formulations deliver the API in a sesame oil vehicle with a small proportion of benzyl alcohol, approximately 1.2%, which acts as a preservative. It is believed to work by blocking dopamine receptors in the brain. We wanted to see how the excipient's purity might impact drug degradation over time. By comparing superrefined sesame oil to standard purity excipients, after 12 weeks at 40 degrees C, the API percentage recovery of haloperidol decanoate was over 93% in superrefined sesame oil. In contrast, two different standard compendial grade sesame oils showed lower API recovery at just 68 and 79% under the same conditions. Again, we observed fewer and less intense degradation peaks with the superrefined sesame oil, confirming greater long-term API stability.
Hi, I'm Chris, Application Team Leader at Croda Pharma. Now, let's take a look at the hormone therapy fulvestrant, which is typically indicated to treat estrogen receptor positive and HER2 negative metastatic breast cancer. Fulvestrant is typically delivered by intramuscular injection and utilises castor oil as a co-solvent and release rate modifier, with many containing greater than 99% weight per volume of oil. Fulvestrant, however, has a known degradation mechanism that's initiated by oxidation of the sulfur atom to form fulvestrant 9-sulfone, as shown. The sulfone formation can be used as an indicator of stability for fulvestrant, typically analysed by HPLC. Fulvestrant is also often formulated in benzyl alcohol, but is also again prone to degradation. Here we compared its stability in superrefined benzyl alcohol versus a competitor parenteral grade. After 12 weeks at 40 degrees Celsius, fulvestrant degraded significantly in the competitor excipient, with nearly 16% sulfone formation, while it remained remarkably stable in our superrefined grade, with levels staying close to zero throughout the study. And when we look at the HPLC chromatograms, you can clearly see the difference. In the competitor excipient, new peaks are present from the outset, specifically benzaldehyde, benzyl benzoate, and then those that form over time, the fulvestrant 9-sulfone. This indicates both the presence of excipient impurities and API degradation. In contrast, formulations with superrefined benzyl alcohol showed clean, consistent profiles with no additional peaks, reinforcing the chemical integrity of both the excipient and fulvestrant. Now, let's take a look at our final API, paclitaxel. Paclitaxel is a chemotherapy drug of the taxane class, used to treat cancers including ovarian, breast, and non-small cell lung cancer. It's often used in intravenous infusion with the originator product Taxol and generic formulations using polyoxyl 35 castor oil at over 50% of the total composition. At Croda, we examined the stability of paclitaxel over 21 weeks at 40 degrees Celsius, comparing compendial grade P35 castor oil, called Etacast 35, with our superrefined P35 castor oil. As shown in the graph, paclitaxel has a 95% recovery under these accelerated conditions, compared with around 83% for the compendial grade material. This further supports that using our higher purity grades of excipient can reduce the risk of degradation of the active ingredient.
Hi, I'm Danila, Applications Team Leader at Croda Pharma. I'll be speaking about our role in supporting the development of generic drug formulations and how we contribute to their success. At Croda Pharma, we are proud to support the development of high-quality generics which serve as essential pillars of affordable healthcare. Generic drug formulations must match the originator in key attributes, but choosing the right excipients can be challenging. Excipient selection is crucial as their interaction with the active pharmaceutical ingredient can influence both product stability and therapeutic efficacy. Even when excipients appear Q1, Q2 compliant, differences in quality and impurity profiles can subtly influence the drug's in vivo behavior and risk profile. Our super-refined excipients are specifically engineered to remove oxidative impurities like peroxides and aldehydes, helping formulators improve drug product stability, enhance shelf life, and minimize risk to bioequivalence. We are trusted by generic manufacturers worldwide not only for our excipients' exceptional quality and consistency, but also for the strength of our global regulatory and manufacturing support. Our portfolio of high-purity excipient products and specialty chemistries solve formulation challenges and enhance drug delivery across a variety of administration routes and dosage forms. Whether it's improved API stability in parenteral formulations where purity is paramount, reducing taste impact in pediatric formulations, or improving capsule performance, our excipients are designed to meet complex formulation needs, ultimately helping our customers accelerate speed to market and deliver better patient outcomes. If you found this video helpful, be sure to subscribe and join us next time on the Croda Pharma YouTube channel as we continue exploring how excipient innovation is shaping the future of pharmaceutical development. Thank you.